Welcome dear students to my channel Biology with Dr. Ishrat. Subscribe my channel and hit the bell icon for latest videos. Today we are going to discuss from chapter number 11 Bioenergetics, and we are discussing the second part of photosynthesis, which is about light independent reaction. So what is light independent reaction? Formerly it was known as dark reaction, but now we will use the term light independent reaction. So dark reaction is also called carbon fixing reaction or C3 cycle. It is light independent process in which sugar molecules are formed from the water and carbon dioxide. The dark reaction occurs in stroma of the chloroplast. As you know that we have discussed in my previous video that chloroplast is the site of photosynthesis in which thylakoids are responsible for light dependent reaction, while stroma is responsible for light independent reaction. So, in the light independent reaction, the stroma of the chloroplast will utilize NADPH and ATP's products of light reaction, and then what will happen, that plant will capture carbon dioxide from the atmosphere through stromata and then proceed for Calvin cycle or C3 cycle or light independent cycle. So in the Calvin cycle, the ATP and NADPH formed during light reaction will convert 6 molecules of carbon dioxide into 1 sugar molecule or glucose. So the chemical equation for light independent reaction is 3 carbon dioxide cycle. 3 carbon dioxide will be used as this reaction, also known as C3, in which your 3 carbon dioxide molecules will utilize, then 6 NADPH will be utilized by adding 5 water molecules and 9 ATPs. Then we will have glyceraldehyde, 3 phosphate and 6 NADP and 9 ADPs and 8 inorganic phosphate will utilize, and this glyceraldehyde 3 phosphate is triose sugar, or plant will use this glucose for its different activities. So what will happen in light reaction? Let's see that this is Calvin cycle, in which you can see that 3 molecules of carbon dioxide, as I have mentioned in my previous slide, that 3 molecules of carbon dioxide will be utilized. Here are 3 molecules of carbon dioxide, and here are the 3 molecules of ribulose biphosphate. Ribulose biphosphate consists of 5 carbon sugar, that's why it is known as ribulose. Ribulose biphosphate, which will add with 3 molecules of carbon dioxide by addition of Rubisco enzyme, and then in first step this carbon dioxide will fixed with the help of Rubisco enzyme and ribulose biphosphate, then they will form phosphoglycerate. So phosphoglycerate is the 3 carbon sugar. You can see that here are the 3 carbons and this is phosphoglycerate, while after that phosphoglycerate by addition of 1 ATP molecules from the ATP, and ATP will convert into ADP. Here are 6 ATP molecules will utilize, and then in the second step, which is reduction of phosphoglycerate into phosphoglyceraldehyde, then NADPH will donate its hydrogen, or this phase is the reduction phase, and then 6 NADPs will form, and this hydrogen ion will add with this phosphoglycerate and it will convert into glyceraldehyde 3 phosphate. So the first step is known as carbon fixation, or you can also call it carboxylation. The first step is carbon fixation, or you can call carboxylation. In the second step, reduction of NADP will occur and then you will have another molecule which is glyceraldehyde 3-phosphate. This molecule is used as a glucose molecules for the plants, and after that this glyceraldehyde 3-phosphate will undergo another phase which is regeneration, in which again 5 molecules of glyceraldehyde will regenerate, and then by addition of 3-phosphate, then again it will convert into ribulose biphosphate. So Calvin cycle will occur during the night time, or it is light independent reaction in which 3 molecules of carbon dioxide will be utilized. It means 2 cycles were consecutively going on with the 3 molecules. This cycle is going on, and other 3 molecules will utilize for the same cycle. Another slide I am showing you that it will also indicate how Calvin cycle proceeds. Here you can see that 3 carbon dioxide molecules and 3 molecules of ribulose biphosphate. Ribulose biphosphate is 5 carbon sugar, so that's why you can see that this is 5 carbon sugar and 3 molecules of ribulose biphosphate will be utilized. And this is the first step. It is known as carbon fixation. By addition of 3 carbon dioxide and 3 ribulose, and by the help of enzyme Rubisco, then 3 molecules of phosphoglyceraldehyde will be formed. Here phosphoglyceraldehyde is triose sugar, and you can see that here are the 6 molecules are formed because of addition of ribulose biphosphate and carbon dioxide. Then what will happen, that 6 ATPs will donate their inorganic phosphate or phosphate group for this reaction to proceed. Then ATPs will convert into ADPs, while on the other hand NADP will reduce and it will give its hydrogen ions to this cycle, and it will convert into NADP. So what will happen, that glycerate molecule, 3-phosphoglycerate molecule will convert into phosphoglyceraldehyde, and this will be the glyceraldehyde 3-phosphate group. Again the same thing, but by the reduction of glycerate into glyceraldehyde. Then what will happen, sugar will form. It means there are 3 carbons are utilized as a sugar, while remaining 5 molecules will again undergo the process of regeneration. Then what will happen in regeneration, that 3 molecules of ATPs are utilized, and then you will have ribulose biphosphate. Overall there are 3 reactions going on in Calvin cycle. The first one is carbon fixation, and the second one is reduction, and third one is regeneration. And I have told you that Calvin cycle will occur in stroma of the chloroplast. Here this cycle is very much clear and indicating that Calvin cycle will proceed by taking 3 carbon dioxide molecules and 3 ribulose biphosphate molecules. Then what will happen, by addition of different enzymes, this process will going on. This is known as Calvin-Benson cycle or Calvin cycle. So again here are 3 steps. What will happen, I will show you the separately what will happen. As you know that Calvin cycle proceed in 3 steps. First step is carbon fixation. In first step what will happen, this ribulose biphosphate molecules is a starting material. It will add with the 3 carbon dioxide molecules, and then what will happen, then 6 molecules of phosphoglycerate will produce, and on its position number 3, carbon number 3 phosphate group is attached. That's why it is known as 3-phosphoglycerate. Then what will happen in the next step, as I have told you that next step is the reduction step or reduction cycle, in which you can see that this 3-phosphoglycerate will convert into glyceraldehyde 3-phosphate, in which 6 molecules of ATPs are utilized their phosphate group, and NADPH will reduce and it will donate its hydrogen, or hydrogen will add in this cycle, and glyceraldehyde 3-phosphate will form. This glyceraldehyde 3-phosphate, its one molecule is used as a glucose or sucrose of the plant. So here are the second step. Then what will happen in third step, in which you can see that 5 molecules of glyceraldehyde 3-phosphate will regenerate by utilizing ATP's energy, and then what will happen? Ribulose biphosphate will form. So there are three steps: carbon fixation, reduction, and regeneration. So here is the again overview of light independent reaction. So let's describe light independent reaction. The first one process is carbon fixation. That plant need the carbon from the carbon dioxide to create the building block for glucose. An enzyme in the stroma called rubisco combined with 5 carbon molecules of ribulose biphosphate with the molecule of carbon dioxide. So this creates a 6 carbon molecule that is broken down into 3 carbon molecules phosphoglycerate. This part of the light independent reaction is referred as carbon fixation. Then the energy carrier from the light dependent reaction make their contribution. As I have told you that ATPs and NADPH are synthesized during the light dependent reaction. So light dependent reaction contribute now, in which ATPs and NADPH will each give phosphoglycerate and a hydrogen atom, creating 2 molecules of simple sugar, which is glyceraldehyde 3-phosphate. So this is the second step, which is also known as reduction. Ultimately, these 2 molecules of glyceraldehyde 3-phosphate are used to build 1 molecule of glucose. This part of the light independent reaction is referred as reduction because reduced to sugar. So the third step, which is regeneration, and it is important to note that the Calvin cycle typically use 6 molecules of carbon dioxide at a time. This means that 12 molecules of glyceraldehyde 3-phosphates are generated. However, only 2 of them are used to produce molecule of glucose, while the rest will recycle back into ribulose biphosphate. So that's why this cycle will keep on running. So here are the goal or the targets of light independent reaction. First is use stored chemical energy to fix carbon dioxide and create a product that can be converted into glucose. Location, as I've told you, that it occurs in the stroma of the chloroplast. Input carbon dioxide, NADPH and ATPs, and output NADH, ADPs, glyceraldehyde 3-phosphate or 2-glyceraldehyde 3-phosphate can be made into glucose. So the ATPs and NADPH thus produced and utilized in the dark reaction or light independent reaction is the formation of carbohydrate. So there is no net gain of ATP in photosynthesis. In the formation of one molecules, 18 ATPs and 12 NADPH molecules are utilized in the 6 turns of the Calvin cycle. Along with that, plant use other pathways like CAM plant and C4. So what is CAM photosynthesis? It is known as crassulacean acid metabolism, because some plants which are present in hot and arid regions, they like cactus and succulent, they close their stomata due to extreme light to reduce the rate of transpiration. So when their stomatas are closed, then what will happen, that carbon dioxide will not enter, so they will use carbon dioxide at night time. Here is the figure, you can see that in night time carbon dioxide will enter the leaf or into the chloroplast. Then this carbon dioxide it utilized by using four molecules or four carbon sugar, which is malic acid, and this will occur in vacuole. So not in stroma, it will occur in vacuole of the cell. So what will happen, that this malic acid is 4 carbon sugar, then this will enter in the chloroplast, then the normal Calvin cycle will occur. So this type of plants are known as CAM plants. Here is the review, that during the hot day their stomata are tightly closed, and so carbon dioxide gas is converted into simple sugar. So during the cooler hours, like in night time, their stomata are open and carbon dioxide enter in the leaf cells, where it combined with phosphoenol pyruvate to form 4 carbon organic acid, which is malic acid and isocitric acid. So the 4 carbon acids are stored in the vacuole of photosynthetic cells of the leaf. So during the daylight hours, the 4 carbon acid break down, releasing carbon dioxide for the dark reaction and for the Calvin cycle of the photosynthesis inside the stroma of the chloroplast. The carbon dioxide then converted into glucose through a series of those reactions which we have already discussed in Calvin cycle. Means these plants utilize carbon dioxide in the night time, when in the daytime carbon dioxide is not reaches to the chloroplast of the cell. Just like this, we have another pathway which is C4 plant. What will happen in C4 plant? These C4 plants combine phosphoenol pyruvate to form 4 carbon organic acid, which is oxaloacetic acid, which migrate to the photosynthetic bundle sheath cell surrounded the vascular bundles of the leaves. So what will happen, that you can see this is the atmospheric carbon dioxide, it will enter into the mesophyll cells. Then what will happen, that these phosphoenol pyruvate molecules will combine with the carbon dioxide, then transport this carbon dioxide for other process like Calvin cycle. So here is the overview, that phosphoenol pyruvate essentially shuttle the carbon dioxide to bundle sheath cell, where it will release for the dark reaction or Calvin cycle. During hot weather, carbon dioxide level inside the leaf is greatly reduced because stomata are closed. The reason that plant want to conserve, or there is a phenomena that plant leaf will conserve water, so their stomata will remain closed during extreme sunlight. So this C4 plants have competitive advantage during hot summer days because they are able to carry out photosynthesis in the bundle sheath, while carbon dioxide levels are concentrated. Weeds like Bermuda grass, purges and porcelain grow during hot summer days. So these are the other pathways than the C3 cycle. You can see that CAM and C4 photosynthesis, because plant will generate their sugar molecules, so they will use alternative pathways. Thanks for watching. For more videos, like, share and subscribe my channel and hit the bell icon so you can update for my latest videos. Thank you so much.